No war in history has changed the human body. But one parasite did. It is smaller than a grain of rice. It has no army, no weapons, and no strategy. It carries a microscopic parasite in its saliva. One bite is enough. Plasmodium, a single-celled organism that invades red blood cells and multiplies inside them until the cell explodes. But over 10,000 years, malaria has killed more humans than every war and famine combined. Some researchers estimate it has killed half of all humans who ever lived. And now, ancient DNA analysis is revealing something even more disturbing. Researchers are now pulling ancient parasite DNA from bones thousands of years old, and what they are finding is rewriting the story of human evolution itself. Malaria did not just kill, it permanently rewrote the genetic code of every population it touched. The numbers were unmistakable. The deadliest blood disorder mapped perfectly onto the deadliest fever. It reshaped the human genome more than any war in history. And it never stopped. In 1949, a British geneticist named J.B.S. Haldane noticed something strange. He was studying a blood disorder called thalassemia. It caused severe anemia, organ damage, and early death. But thalassemia was not random. It appeared at highest frequency in exactly the places where malaria was most deadly: Italy, Greece, Sardinia, the coastal lowlands of Turkey, every malaria belt in the ancient world: Sardinia, Greece, coastal Turkey, every lowland where warm, stagnant water collected. The list grew longer with every study. Carried the highest rates of a disease that should have been lethal. Haldane asked the obvious question: why would evolution keep a deadly gene? His answer became one of the most important ideas in human genetics. Haldane proposed that thalassemia survived because carriers, people with one copy of the mutated gene, gained protection against malaria. 2 copies killed you. 0 copies left you exposed to malaria, but 1 copy gave you just enough resistance to survive both. 70 years later, whole genome sequencing proved Haldane right, and revealed the scale was far larger than anyone imagined. Malaria did not just select for one mutation. It drove at least 6 separate genetic changes across 3 continents, each one arising independently in populations that never met. 6 separate mutations, 3 continents, each one a different genetic answer to the same lethal question. And in populations separated by oceans and millennia, evolution converged on remarkably similar solutions. The entire malaria belt, rewritten at the genetic level. The same parasite forced the same evolutionary answer in completely different people. The most famous example is sickle cell. A single mutation in the hemoglobin gene changes the shape of red blood cells from round discs to rigid crescents. 2 copies cause sickle cell disease, organ damage, pain crises, shortened life. But 1 copy makes red blood cells inhospitable to the Plasmodium parasite. Carriers survive malaria at dramatically higher rates. In sub-Saharan Africa, where Plasmodium falciparum kills hundreds of thousands of children every year. Today, roughly 300 million people worldwide carry at least 1 copy of the sickle cell gene. In some West African communities, 1 in 4 newborns carries the trait. 5 separate events, 5 populations that never exchanged a single gene. The sickle cell gene reaches frequencies of 40%. 40%. That is not a rare mutation. That is a population-wide genetic rewrite. And then researchers discovered sickle cell had not evolved once. It had evolved at least 5 separate times. 5 independent origins: the Benin haplotype, the Bantu haplotype, the Senegal haplotype, the Cameroon haplotype, and the Arab-Indian haplotype. Each one arose in a different population on a different continent, thousands of years apart. The same single nucleotide change in the same gene, invented 5 times by 5 unrelated populations under the same pressure. That is convergent evolution, the strongest proof that malaria is the most powerful force our species ever faced. Evolution repeated itself because the pressure left no other option. But sickle cell was only the beginning. The parasite forced other changes too. Glucose 6-phosphate dehydrogenase deficiency, G6PD. It is the most common enzyme deficiency in the world. Roughly 400 million people carry it. And like sickle cell, it appears at highest frequency in malaria endemic regions. The enzyme normally protects red blood cells from oxidative stress. Without it, certain foods and medications can trigger a hemolytic crisis, destroying red blood cells rapidly. But that same fragility makes the cells a hostile environment for Plasmodium. In the Mediterranean, the condition was historically called favism. Eating fava beans could trigger a crisis. Entire villages knew to avoid them, but no one understood why. The connection to malaria would not be made for another century. Sardinia has one of the highest G6PD rates on Earth. It also had some of the worst malaria in Mediterranean history. The pattern repeated. Wherever malaria killed the most, humans evolved genetic defenses that came at a brutal cost. And then there is the Duffy antigen, the strongest selective sweep ever measured. One protein, one doorway. The parasite needed it to survive. The Duffy antigen is a protein on the surface of red blood cells. Plasmodium vivax, one of the 2 deadliest malaria species, uses it as a doorway to enter the cell. In sub-Saharan Africa, a mutation arose that silences the Duffy gene entirely. No protein, no doorway, no infection. Today, 97% of people in sub-Saharan Africa carry this mutation. 97%. That means a single parasite drove a genetic change so powerful that it reached near total fixation across an entire continent. No other known genetic change in human history reached this level of fixation this quickly. Population geneticists call it the strongest known selective sweep in the entire human genome, stronger than any adaptation to climate, diet, altitude, or sunlight. But here is the twist. These mutations were never free. Sickle cell disease kills over 300,000 children every year, almost all of them in Africa. When 2 carriers have a child, there is a 1 in 4 chance the child inherits 2 copies. 2 copies is not a survival advantage. It is a death sentence. G6PD deficiency triggers hemolytic crises from common foods and medications. Thalassemia major requires lifelong blood transfusions. The World Health Organization estimates that 75% of sickle cell births occur in sub-Saharan Africa. Many die before diagnosis. Beta thalassemia major requires transfusions every 2 to 4 weeks for life. These are not theoretical costs. They are measured in children who die before their 5th birthday, because the same gene that protects them from malaria destroys them from within. Evolution does not care about individuals. It cares about populations surviving. In populations where malaria killed 1 in 5 children, any gene that improved survival spread rapidly. Geneticists call it balanced polymorphism, the cruelest form of natural selection, regardless of what it did to the unlucky minority who inherited 2 copies. The math is merciless. If malaria kills 20% without the trait, sickle cell kills 25% with 2 copies. The math still favors keeping the gene. The population wins even as individuals lose. And that mathematical truth is now written permanently into the genomes of billions of people alive today. In 2016, a team at McMaster University extracted ancient DNA from 2,000-year-old skeletons buried across the Roman Empire. They were looking for Plasmodium falciparum DNA, genetic material from the malaria parasite itself, preserved inside ancient human bones. Ancient pathogen DNA degrades rapidly. It fragments into tiny pieces. Extracting it from contaminated bone demands extraordinary precision. They found it. Plasmodium falciparum DNA in individuals buried in Roman cemeteries in Italy, dating to the 1st and 2nd centuries. Ancient Rome had malaria, and the genetic evidence was still inside the bone. Roman writers described a deadly fever haunting the marshlands around Rome. They called it malaria, bad air. They did not know a parasite transmitted by mosquitoes was responsible. But their bones recorded the truth. The Pontine marshes south of Rome were notorious. Entire legions refused to camp near them. Augustus spent a fortune draining the marshes. Within a generation, they flooded again. Roman generals knew the marshes killed soldiers. They just blamed the air, not the mosquitoes. And the genetic signature of that ancient malaria matches the strains that still circulate in tropical Africa today. An earlier study by Robert Sellers had detected malaria DNA in Roman era remains from Lugnano in Teverina, a 5th century infant cemetery in Umbria. The infants buried there showed signs consistent with severe malaria infection. They had been placed in graves with protective amulets, raven claws, toad bones, and bronze tokens meant to ward off the disease their parents could not name. Malaria shaped European genomes too. The thalassemias Haldane noticed in Mediterranean populations are the direct evidence. And the timeline matches. Beta thalassemia mutations in the Mediterranean date to roughly 8,000 years ago, precisely when agriculture expanded into marshy lowlands and created the standing water that mosquitoes breed in. Every irrigation ditch was a breeding pool. Every rice paddy was an incubator. The mosquito population exploded alongside the human population, and the parasite followed both. Mesopotamia, the Nile Delta, the Ganges floodplain. Every cradle of civilization was also a cradle of malaria. Humans created the conditions. The parasite did the rest. When early farmers cleared forests and built irrigation channels across Mesopotamia, the Nile Delta, and Southeast Asia, they built paradise for Anopheles mosquitoes, warm, shallow, stagnant water surrounded by dense human populations. Malaria exploded, and so did the genetic mutations that fought it. The Duffy antigen tells an even older story. The mutation reached high frequency in Africa roughly 30,000 years ago. That predates agriculture by 20,000 years. It means malaria was already killing humans in large numbers during the Stone Age. This was not a disease that arrived with farming. It was already ancient. Plasmodium vivax, the malaria species that uses the Duffy antigen, was already circulating among early human populations in tropical Africa, and the selection pressure was so intense that it drove a genetic change to near total fixation across an entire continent before the first grain of wheat was ever planted. The same pattern repeated in Southeast Asia. Alpha thalassemia and hemoglobin E variants reached high frequency in populations across Thailand, Cambodia, Laos, and Indonesia. In parts of Thailand, hemoglobin E carrier rates reach 40%. Same parasite, same lethal bargain. 3 continents, 6 mutations, 1 parasite. Each population evolved its own answer to the same question. Screening programs now test every newborn. The mutations are too common to ignore. The evidence now spans decades, hundreds of studies, and millions of genomes. The conclusion is unavoidable. In 4 hours. So what does this actually mean for how we understand human evolution? Textbooks describe human evolution as shaped by climate, diet, migration, and conflict. Ice ages selected for cold tolerance. Agriculture drove lactase persistence. Altitude selected for efficient oxygen transport in Tibetan and Andean populations. Across every chromosome, the same story. Malaria left its signature everywhere. But when researchers scan the human genome for the strongest signals of natural selection, the largest, fastest, most dramatic sweeps. Malaria resistance genes dominate the list. They are not just present, they are the largest signals in the entire dataset. No famine, no war, no ice age left a bigger mark on our DNA. A single mosquito-borne parasite reshaped the biology of our species more profoundly than every army that ever marched. And the process is not over. Malaria still kills over 600,000 people every year, mostly children under 5 in sub-Saharan Africa. New studies identify previously unknown protective mutations every year. The catalog keeps growing. African genomics programs are now sequencing populations that were never studied before and finding malaria signatures everywhere they look. The selection pressure continues. New protective variants are still being discovered. The genome is still being written. Each new variant tells the same story. The parasite attacks, the genome adapts. The cost is paid in blood. In 2023, a genome-wide association study across multiple African populations identified new protective loci that had never been cataloged before. The war between humans and malaria is still being fought inside our chromosomes. Modern medicine fights malaria with bed nets, insecticides, and anti-malarial drugs. A vaccine called RTS was approved in 2021, but the genetic legacy remains. Sickle cell trait, thalassemia, G6PD deficiency, and Duffy negativity are not going away. They are permanent features of the human genome, written by a parasite editing our species for 30,000 years. Every child born today in a malaria endemic region inherits a genome shaped by this ancient arms race. Some carry sickle cell trait, some carry G6PD deficiency, some carry both. Their genomes remember what their textbooks forgot. If you found this useful, subscribe. We cover the DNA evidence behind every major historical claim. The question that remains is not whether malaria rewrote the human genome. It did. The evidence is overwhelming. The question is, what other parasites, what other invisible pressures are still writing our genetic code right now? And what will the genome of our species look like 1,000 years from now? If history is any guide, the answer will not come from our choices. It will come from what hunts us.